Welcome to my OCR A-Level Biology revision session with me, Christine. So in this lesson, I'm going to look at classification and the domain system. So what is classification? Well, classification is the way in which we group organisms together. So we group them, and if somebody is going to study the principle of classifications, then what they're studying is taxonomy, because what we're looking at is the taxons. Now, a group of one or more populations of organisms seen as a unit by a taxonomist is what we know as being a taxon. So when we group them together, we're going to group them into domain, kingdom, phylum, class, order, family, genus, species. Now, I like to do things using the alphabet, so I always try to remember this by KPC or FGS. Kingdom, phylum, class, order, family, genus, species. Now, domain goes above that, and I'll talk about when the domain system come in. However, what's important that we note is at the domain level, this group is the largest group. All the organisms that are in the domain taxon have the smallest number of similarities. So therefore, they share the ancestor which was at the beginning. So there is a distantly shared ancestor. However, they're still an ancestor. They just have the smallest number of similarities. Now, when we work our way down through the taxons, from the kingdom to the phylum to the class to the order to the family to the species, missed out genus there, genus then species, what we get to is the smallest group. So the smallest group is the species level. This is the one that has the largest number of similarities, and therefore the smallest group of organisms are held within this species. We therefore say that they are the most recent common ancestors. Now, remember, a species is a group of individuals within a population who are able to interbreed and produce fertile offspring. So it's important that we note that these individuals, when they reproduce, they can produce fertile offspring. So if we look at the different versions that we could have with regards to different organisms. So if we're looking at classifying a human or a chimpanzee or the meadow buttercup, we will see that they are all within the same domain. They are all within the domain of Eukarya. As we move down through the taxons, we will start to see that they separate out. And that's important because that is the way in which we classify organisms, is by grouping them. So we can see the humans and chimpanzees are grouped together in the kingdom of Animalia, whereas the meadow buttercup is in the kingdom of Plantae. So that's already been separated out when we were grouping the organisms. Now, the human, the chimpanzee, the meadow buttercup, these are what we call the common names for these individuals. But what you have to understand is me being English, well, Scottish, but speaking the English language means that I will say something one way, whereas if it was said in another language, it may be translated to something else. And therefore, what we do is we use what's known as the universal language. And we do this by doing a binomial naming system. So we do this binomial nomenclature, and that is where we use the generic name as the first name. So that is our genus. We always write this with a capital letter. Then we move on to the specific name, the lowercase, and that is the species name. So whenever we name an organism using the scientifically correct way, we use the generic name and then the specific name. So how could they ask a question about this in the exam? Well, what they will do is they will give you some hints. So here we have the European badger. They're told the student that it's within the same member of a family. So remember that KPC or FGS, kingdom, phylum, class, order, family, genus, species. So they're within the same family. So now we have to understand, well, if they're in the same family, they're either going to be in the same genus because it's their generic name of the badger, but in a different species level, So therefore we would expect that first name to be the same. And so we can see at the top, the European badger is Meles. So therefore we would expect the generic name to be Meles and the specific name to be, for the American badger, something different. So that shows us that the first one is the name of the American badger. So we are being asked to apply our knowledge to a random situation. And they've also given us some hints here, because look at where you've got your capitals and your lowercase. So only the top and the bottom actually follow the correct rule of how the naming should be written. Obviously, if it's typed, it's much easier to do it in italics. If you are writing this in the exam, there is an expectation that you would underline, because you cannot show italics very easily when you're writing. You therefore underline instead. Another way they could ask the question is they actually give you the polar bear and the giant panda. They've told you that they belong to the same family. Do they belong to the same class? Well, again, kingdom, phylum, class. Class is above, order, family. Therefore, they would be in the same class if they are in the same family, because they have to be grouped together. So as it moves down through the taxons, the groups get smaller and smaller and smaller. So if they're in a family together, that means everything above, they are the same. Okay, so those kingdoms then, there are 5 kingdoms that you have to be able to distinguish between: prokaryotes, protoctista, animalia, plantae, and fungi. So let's go through and look at each one individually and see what are the distinguishing features for them. So let's start with the prokaryotes then. So prokaryotes are our prokaryotic cell. Now, the prokaryotic cells, remember, have got that circular DNA. It's naked. It's not held in a nucleus. There's no histone proteins associated. So when we talk about prokaryotes and we are distinguishing and classifying them, these are the features that we're looking at. Now, prokaryotes are unicellular. They are small, one-celled organisms. So that is a classification that we are going to be looking at. If we found an organism that's unicellular, we will start to think, well, if it's unicellular, does that make it prokaryotic? If we then look at the cell structure, that's when we will start to distinguish between whether it's prokaryotic or potentially one of the other kingdoms. Now, one of the key things about prokaryotes is that they do produce extracellular enzymes. So they are going to produce enzymes that are excreted out of the cell that will do the digesting externally. Then what they're able to do is absorb nutrients from their surrounding environment. The other thing they can do is they can also produce their own nutrients by photosynthesis. So if they have the pigments, they have chlorophyll, they can then absorb the light energy and they are able to then produce those nutrients, and that makes them autotrophic. So examples of these are bacteria or cyanobacteria. Now let's move on to the protoctistas then. So protoctistas, these are one of the eukaryotic organisms. So these are eukaryotic, which means that they have got their nucleus, they have their membrane-bound organelles, their DNA is associated with histone proteins. So this is linking back to module 2 and that cell structure, the distinguishing features between whether it's prokaryotic or eukaryotic. Now, protoctistas are mainly unicellular. So if we were looking at a cell and we were trying to identify it, we'd start off with, if it's unicellular, is it a protoctista or is it a prokaryote? And that would be where we start to look at those structures inside the cell. Now, protoctistas can be autotrophic. Now, auto means self, trophic means the way in which they feed. So if they are a autotrophic feeder, that means that they are self-feeding, which means they're doing things like photosynthesis. So some of them could be autotrophic, which means they are self-feeding. So they may be using light energy and converting that into organic compounds, or it could be that you can use chemicals. So an autotroph can feed itself because it's able to make its organic compounds that are necessary for respiration. If an organism isn't able to make its own nutrients, what it needs to do is it needs to feed on another. And we say that they are heterotrophic if they must feed on something else to get the organic compounds that they require. So protoctistas can either be autotrophs or heterotrophs, or they can actually be both depending on their environment. Now, some of them are parasitic, and you'll learn about that when you do communicable diseases and you look at malaria, which is an example of a communicable disease which is caused by a protoctista. So examples of things like this is algae that you can see in my picture, or protozoa, which is the one that causes malaria. Okay, let's move on to fungi then. So fungi are eukaryotic as well. So we know that they are eukaryotic, so they have all those membrane-bound organelles. Now, fungi can either be unicellular, one cell, or they can be multicellular. So this is our way to distinguish between those that are unicellular, fungi, protoctista, prokaryotes, and now if we want to classify an organism away from that, if they are multicellular, we know that they are potentially fungi if we were only looking at those three kingdoms as our example. So unicellular or multicellular, so they can be either or. Now they actually get their nutrients by absorption. Now they, like prokaryotes, are saprotrophic. So what the fungi does is the fungi excretes the digestive enzymes like we were talking about with the prokaryotes, and they then absorb those broken down nutrients from the external environment. They absorb that back in. So they're normally found on food that is decaying. So you can quite clearly see where you have I say quite clearly see, but I have a picture here of a brownie, which unfortunately I had left for a little bit too long, and then I got excited by how it looked in its mold state as it was growing. So the fungi, is important to note that they are eukaryotic. They store their food as glycogen. This is a new distinguishing feature for them, in the fact that compared to the prokaryotes and compared to the protoctistas, when we talk about fungi, because they can be multicellular, as they get bigger, they need a way of storing that food. So if they need to store it, they store it as the polysaccharide glycogen. The other thing to note with fungi is that yes, they're eukaryotic with a cell wall. So we can distinguish the cell wall, therefore distinguish fungi from an animal because they have the presence of a cell wall. However, the cell wall is made up of chitin, which is a distinguishing feature to be able to distinguish it from a plant which has cellulose. So mushrooms, yeast, mold are all examples of fungi. So plantae then, another eukaryotic organism, multicellular, never unicellular. So plantae, within the plantae kingdom, these are multicellular organisms. They're made up of many cells. Their cell wall is made up of cellulose. So that would then link us back to our polysaccharides and the fact that it's a beta glucose which is going to be joined together, creating our cellulose fibers. They are autotrophic feeders. So plants do photosynthesis. They have the necessary pigments to absorb the light energy so that they can do photosynthesis. They store their food as starch. So remember, going back to the polysaccharides, starch is made up of amylose and amylopectin, and that is how the plant stores its food for when there might not be, for example, enough light for that photosynthesis. So they can produce the nutrients, those organic compounds, but then they need to store them for a later date so that they can then use them. So there's lots of different examples for plants, your mosses, your ferns, your flowering plants, your conifers. All of these are grouped within the kingdom of plantae. So Animalia then, I mean, this is huge. Animalia is eukaryotic, again, multicellular. So we have established that protoctistas and prokaryotes are only unicellular. The minute we get into a multicellular organism, we automatically can remove them from our classification grouping, and now we're focusing only on the three: fungi, plantae, and Animalia. They are all eukaryotic, so we know they have membrane-bound organelles, a nucleus, and DNA, which is associated with histone proteins. This time, however, Animalia are not able to self-feed. So because Animalia can't do the self-feeding, they are heterotrophic feeders. They have to feed on the producers, which are the plants, or they have to feed on other organisms that feed on the plant, so they are consumers. They also store their food as glycogen, so it's important to note that they also store that food as the polysaccharide, and there's so many different examples that you can have for the Animalia classification. So we know that we have our 5 kingdoms, and our 5 kingdoms are a way in which we can classify organisms. In 1990, Carl Woese actually used results from studying ribosomal RNA. Ribosomal RNA is necessary, remember, for making the polypeptides that are produced. He also looked at the cell membrane lipid structure in different organisms and the effect that antibiotics had on organisms to distinguish them into 3 distinct groups. So we have the first one, and this is what we call our domain system. So eukarya, we can distinguish them into any organism within this domain if they have ATS ribosomes. So their ATS ribosomes is a distinguishing feature that puts them within the eukarya domain. So that therefore means all of our eukaryotes. We know that eukaryotic cells have ATS ribosomes, therefore they would be classified within that eukaryote domain. So we can see our protoctista, Animalia, plantae, and fungi, and you can see some distinguishing features. Are they unicellular? Are they multicellular? Do they have a cell wall? Do they not have a cell wall? What is that cell wall made out of? And what is their feeding procedure that they do? The other thing we can look at is the RNA polymerase. So remember, for transcription to occur, RNA polymerase is needed. Well, in RNA polymerase, we have the 18 proteins involved. So this is important for us to note that RNA polymerase has those 18 proteins. Well, now what we need to do is look at the bacteria. So we know that prokaryotes are bacterial cells. Well, they've got 70S ribosomes. Their cell wall is also made up of a different substance. So if there's a cell wall present, this is a way to distinguish between whether it is a bacterial cell compared to a plant cell and a fungi cell based on what is that cell wall made up of. So this is made up of peptidoglycan, which is a distinguishing feature. Its RNA polymerase is actually only made up of 5 proteins. So therefore, we have our 5th kingdom, which is prokaryote. Well, actually, Carl Woese, when he started to do this research, he realized that the prokaryotes needed to be separated into 2 different domains, because although they all have 70S ribosomes, their cell wall is actually made up of different substances. So therefore, that is a distinguishing feature as to whether it is a bacteria, true bacteria, or whether it is what's known as an archaean bacteria. And that is one of the more extremophiles in which they can withstand extreme conditions. So they have a different type of cell wall made up of non-murin, and their RNA polymerase is actually made up of 8 to 10 proteins. So by doing this research and by advancement of technology, what we've actually managed to do is take our 5 kingdoms and separate them out into 6 kingdoms. Are they eubacteria, the true bacteria that we find in our everyday lives, or are they archaean bacteria, ancient bacteria, who have a common ancestor with the eukarya. So it's really important when we look at classification that we understand that classification is always evolving as we have more and more evidence and more information about the different organisms. But what you have to be able to do is you must be able to describe how the classification system is used and also the difference between the 5-kingdom system and the 6-kingdom system with the 3 domains. So it is important you note that there are different scientists, some who still use the traditional prokaryotes and the eukaryotes, for eukaryotes, 1 prokaryote, and those who actually do split it up into a 6-kingdom with the 3 domains. So if you like this video, then please do click on the like button and subscribe to my channel. Also, check out my revision platform, www.aiqchat.com, to help you with your revision.